for all the incredible progress in understanding biology, digging into the spatial organization of things where molecules and cells are within tissues has felt like this incredibly intricate puzzle, often requiring, well, just a ton of manual computational work.
Mm.
But it feels like we're on the cusp of a real shift, wouldn't you say?
Oh, absolutely.
And that's exactly what brings us to today's deep dive.
Yeah, we're seeing a fascinating evolution where
Mhm.
AI is moving beyond just processing data to actually becoming an active participant in the scientific process itself.
Right.
And at the forefront of this, I think, is the emergence of these fully autonomous AI agents for research.
Which leads us straight to our main topic today, Spatial Agent. It's generating quite a bit of buzz, actually. It's pitched as a fully autonomous AI agent designed specifically for tackling the complexities of spatial biology research.
That's the one.
So, for you listening, our goal today is to really get under the hood of Spatial Agent. What is it? How does it pull off these impressive feats? And why, maybe, its arrival could be a game changer for the field.
And to do this, we'll be focusing pretty closely on the research paper that actually introduced Spatial Agent. Think of this as your guided exploration. We'll look at the core ideas, the innovative technology, and of course, the key findings straight from the source.
Right. So we're going to start by laying the groundwork, the why behind Spatial Agent, then dive into the ingenious tech that makes it tick, explore the significant results and what they tell us, and finally, understand why this all has such important implications. Okay, let's start by setting the stage. Where does Spatial Agent come from in the context of scientific research?
Well, the paper really kicks off by emphasizing the growing, I'd say, transformative role of artificial intelligence in scientific discovery. It's hard to miss.
Definitely.
We've seen really remarkable progress recently with large language models, LLMs, showing they can, reason through complex problems, devise plans, and even seamlessly integrate different computational tools.
Yeah.
And the logical next step in this evolution seems to be the development of these autonomous agents.
Autonomous agents. So this is more than just like running pre-programmed analyses.
Precisely. Yeah, the research paper defines autonomous agents as AI systems that have the capacity to perceive their environment, which in this case is scientific data, and tools formulate plans to achieve specific goals and then act on those plans iteratively.
Iteratively.
Right, and all with minimal need for human intervention. It's almost like having a dedicated research assistant that can independently navigate the complexities of a spatial biology project.
That sounds like a fundamentally more flexible approach than how things are often done, traditionally.
Exactly. The paper draws a pretty clear distinction between these agents and, say, traditional automated pipelines.
Okay.
Think of those pipelines as very specific, sort of hard-coded sets of instructions. They're great at repetitive tasks with well-defined parameters, but they lack that crucial adaptability, right? They struggle with the inherent variability of new biological data or, unexpected research questions.
Yeah, biology's messy.
It is. And then, of course, there's the conventional human-driven analysis, where a researcher has to manually select tools, interpret outputs, constantly apply their own deep biological knowledge. Autonomous agents are kind of aiming to synthesize these approaches.
So it's trying to capture the tireless efficiency of automation, but with the, let's say, intellectual flexibility of a human expert.
That's the core idea, yeah. And the paper even points to some recent examples where autonomous agents are already showing their value in various scientific areas, things like refining research hypotheses, automating complex molecular simulations, even assisting in therapeutic drug discovery.
Okay, so we get the rise of AI agents in science generally. Now, why focus this powerful tech specifically on spatial genomics? What's so special about that field right now?
That's a critical question. Spatial genomics is, well, it's a really dynamic and rapidly expanding field. It's all about understanding that intricate spatial organization of biomolecules and cells within the context of tissues.
Right, the where matters.
Exactly. And this spatial information is proven to be absolutely vital for understanding a whole range of biological processes, from how our bodies maintain a healthy state homeostasis to what goes wrong in various diseases.
Makes perfect sense. I mean, where things are located and how they interact spatially must be just as crucial as knowing what those things are.
Absolutely.
But the paper also points out some unique hurdles in spatial genomics research.
Yes, because it's a relatively young discipline, the computational methods are still quite fragmented. We haven't really seen the emergence of standardized, universally applicable analysis pipelines yet. This often means a significant amount of manual effort and expert interventions needed just to piece together different analytical steps. Plus, methods that work well on one type of spatial data set might struggle or even fail completely on another. There's a real challenge in achieving broad generalization.
Right.
And fundamentally, interpreting spatial genomics data requires a deep, integrated understanding of biology at multiple levels, from molecules up to cells within their tissue environment.
So to truly make sense of this data, you practically need to be an expert in multiple fields at once.
It can certainly feel that way. And that's precisely where the central concept behind SpatialAgent comes into play. The researchers basically hypothesize that an LLM-driven autonomous agent could be ideally suited to navigate this intricate landscape.
Okay, so what's the core innovation with SpatialAgent then? What is it actually designed to do in the realm of spatial biology?
The fundamental idea is that SpatialAgent is designed to autonomously manage the entire spatial biology research workflow. And that means starting right from the initial experimental design considerations, moving through the complex data analysis steps, and even extending to generating novel research hypotheses.
Wow, that's a pretty comprehensive scope. What makes it stand out from just, say, using a collection of existing software tools and databases?
According to the paper, the key differentiators lie in its capacity for adaptive reasoning and its ability to dynamically integrate a diverse set of tools.
Adaptive reasoning.
Yeah, unlike traditional computational approaches that follow rigid, predefined workflows, SpatialAgent can intelligently adjust its analytical strategy. It does this based on the specific characteristics of the data it sees, the type of tissue being looked at, and the particular biological questions being asked. It's not sort of locked into a fixed sequence of operations.
So it has a level of computational intuition almost.
In a computational sense, yeah, you could say that. It's designed to handle various forms of input data, what the researchers call multimodal inputs. It can proactively access and use external databases for relevant information. And it even incorporates mechanisms for researchers to step in and provide guidance or refine the process as needed. That's the human-in-the-loop capability they mentioned.
And this wasn't just a theoretical framework, right? They actually put SpatialAgent through some pretty rigorous testing.
Oh, absolutely. The research paper details extensive evaluations. They tested SpatialAgent's performance using real-world spatial biology datasets from the human brain, the human heart, and also a mouse model of colon inflammation. And it was tasked with performing a range of critical analyses, like designing optimal gene panels for targeted studies, annotating different cell types within tissues, and inferring underlying spatial patterns of cellular organization and communication.
And by all accounts, the initial results were quite encouraging.
Indeed, the paper reports that SpatialAgent not only performed competitively, but in many instances, actually significantly outperformed existing state-of-the-art computational methods. It even matched, or in some cases, surpassed the analytical capabilities of human experts on several key spatial biology tasks.
Okay, that's a compelling overview of what SpatialAgent aims to achieve. Now let's really dig into the nuts and bolts. How does this innovative technology actually work? What's under the hood that enables it to perform these complex analyses?
Right. So at its core, SpatialAgent is described as an LLM-based agent. It operates through what the researchers call a self-governing loop. This loop is really the fundamental mechanism that allows it to seamlessly integrate the powerful reasoning capabilities of large language models with a diverse array of external computational tools and biological knowledge databases. Figure 1A in the paper gives a pretty helpful visual of this architecture.
A self-governing loop? That sounds quite sophisticated. What are the key components that make up this loop?
Okay, so the agent is equipped with what they term predefined planning templates. You can think of these as being kind of analogous to the established protocols or maybe informal best practice guides that experienced bioinformaticians often rely on for common analysis tasks.
Ah, like playbooks.
Exactly, playbooks. These templates provide a structured starting point for tackling frequently encountered problems in spatial biology. But crucially, SpatialAgent also has access to a carefully curated and extensive set of domain-specific computational tools. The researchers really emphasize that providing the LLM with the right specialized tools is absolutely essential for it to perform effectively and accurately in a complex scientific domain like this.
So it's not just a general-purpose AI trying to decipher biological data. It has a specialized toolkit tailored for spatial analysis.
Precisely. And importantly, SpatialAgent isn't a static system. The paper highlights its ability to dynamically adapt its behavior. It can adapt in response to feedback from human users, it can incorporate new tools as they become available in the field, and it can even tackle novel analytical tasks that it hasn't encountered before. Supplementary figure 8 in the paper illustrates this adaptability quite well.
Okay, so it has these planning blueprints and a dynamic set of tools. How are these organized within SpatialAgent itself?
So the researchers detail three key interconnected modules that form the core of SpatialAgent's architecture. These are the memory module, the planning module, and the action module. Their interactions are nicely laid out in figure 1b of the paper.
Memory, planning, and action. Those sound like the fundamental requirements for any intelligence system, really. Let's start with the memory module. What kind of information does SpatialAgent retain and use?
Right, so the memory module is responsible for maintaining two distinct but complementary types of information. First, it holds what's called semantic memory. This covers the high-level goals provided by the user, plus detailed knowledge about the various computational tools available to the agent and, what they do.
Okay.
For instance, it might store the user's objective, like identify key marker genes for specific cell types in a mouse brain tissue section, along with info on how to use different single-cell RNA sequencing datasets and marker gene databases.
Yep.
Second, it maintains episodic memory. This records the short-term steps it has taken during the current analysis, the intermediate results it's obtained, and the evolving context of the ongoing task. This basically ensures the agent can effectively track its progress within a complex workflow.
So it remembers both the overall objective and the specific actions it has taken to get there. That seems crucial for maintaining coherence in a potentially lengthy analysis.
Absolutely essential.
What about the planning module? How does SpatialAgent decide on the sequence of steps to take?
The planning module is where the agent's reasoning capabilities really come into play. It leverages chain-of-thought reasoning, which is a technique that allows the LLM to break down complex, multi-step tasks into a logical sequence of smaller, more manageable sub-goals. It also uses self-reflection prompts. These are specifically designed prompts to encourage the LLM to critically evaluate its own plans, maybe identify potential pitfalls or inconsistencies, and then refine its strategy as needed. The supplementary information in the paper goes into more detail on these prompting techniques.
Self-reflection for an AI. That's a fascinating concept. Does it always generate these plans from scratch, or does it have some pre-existing strategies?
Well, the planning module has the flexibility to operate in both ways. It can optionally use those predefined planning templates we talked about earlier, the analyst playbooks, which offer a ready-made structure for common spatial biology tasks.
Okay.
However, it also has the capability to generate analysis plans de novo entirely from scratch if a suitable template isn't available for a given novel research question. And, maybe most importantly, a key aspect of the planning module is its ability to dynamically refine its plans as the execution progresses and new information becomes available. This could be in response to the results of a particular tool, or maybe due to changes or clarifications from the human user.
So it's not just rigidly following a preset path. It can actually adapt its approach based on the evolving situation.
Exactly. Which brings us to the final piece of this core architecture, the action module. This is where the carefully constructed plans are actually translated into computational operations.
Makes sense. What kinds of actions are we talking about here in the context of spatial biology research?
The action module is responsible for selecting and executing the appropriate computational tools to carry out each step in the plan. This could involve a really wide range of operations, like retrieving relevant biological data from online databases, think single-cell RNA, sequencing repositories, or performing cross-species gene name conversions to make sure datasets are compatible, or maybe verifying potential ligand-receptor interactions using specialized knowledge bases.
Right. Lots of different steps.
Yeah, and the action module can also process data using established scientific computing libraries that are widely used in the field, like scikit-learn for machine learning, PyTorch for deep learning models, and Scampi for single-cell transcriptomics analysis. But importantly, it even has the capability to generate and execute custom Python code when necessary to perform specific or novel analyses. And the researchers emphasize that the toolbox of available actions is designed to be easily extensible. Users can integrate new tools and functionalities to tailor SpatialAgent to their specific research needs. The supplementary info has more on that extensibility.
So it can not only access existing tools, but also create its own if needed. That sounds incredibly versatile.
It is. Now, let's talk about how researchers actually interact with and use SpatialAgent. The paper describes two main modes of operation: autonomous mode and co-pilot mode.
Autonomous mode sounds like it requires minimal user input, right?
That's correct. In autonomous mode, Once a researcher provides an initial high-level query or research objective that aligns with one of those predefined plan templates, SpatialAgent can execute the entire complex analysis workflow from start to finish without any further human intervention. It's designed to be able to generalize its approach to unseen requests, as long as they fall within the scope of those established templates.
So for well-defined and common analysis tasks, you can essentially set it and forget it. Sort of.
That's the underlying principle for autonomous mode, yeah.
Huh.
But what if the research question is more exploratory? Or if a researcher wants to actively guide the analysis process?
Good question.
That's where the co-pilot mode comes in. This mode enables a much more interactive and collaborative engagement between the researcher and SpatialAgent. Users can actively refine the task definition as the analysis unfolds, maybe by asking follow-up questions to explore specific findings in more detail, or by providing additional instructions to steer the agent's focus.
Ah, okay.
This interactive capability makes SpatialAgent highly adaptable for tackling complex or novel research questions, where the initial direction might actually evolve as new insights emerge.
So it can act as a highly intelligent research assistant that you can directly collaborate with throughout the entire process.
Precisely. And to thoroughly evaluate the effectiveness and versatility of SpatialAgent, the researchers conducted extensive benchmarking across those three key use cases we mentioned earlier: designing gene panels, annotating cell types and tissue niches, and mining data patterns to generate hypotheses. These evaluations involve using three different predefined plan templates, one for each use case, a comprehensive set of 19 specialized computational tools, and a diverse collection of 5 different spatial biology datasets.
That sounds like a very thorough evaluation setup. So what were the key findings across these different use cases? Did SpatialAgent demonstrate the capabilities the researchers were hoping for?
Let's start with the gene panel design use case.
Hmm.
Designing an effective panel of genes is absolutely critical for these targeted spatial experiments, right?
Crucial, yeah.
So the researchers rigorously compared SpatialAgent's performance against several well-established computational methods currently used, and even against gene panel designs created by 10 human experts, people with significant experience in spatial transcriptomics. They used a detailed spatial transcriptomics dataset of the human dorsolateral prefrontal cortex for this.
And how did SpatialAgent's designs stack up against the existing algorithms?
The paper reports that the gene panels designed by SpatialAgent consistently outperformed those generated by the computational baseline methods. They saw significant improvements in key metrics like cell type prediction accuracy ranging from 6.0% to 19.1% higher, and also substantial gains in the accuracy of predicting the spatial coordinates of cells, with R-squared value increases up to 47.1%.
Wow, those are pretty significant improvements over existing computational approaches. What about compared to the intuition and expertise of human scientists?
Well, interestingly, SpatialAgent's gene panel designs actually outperformed 90% of the panels created by the human experts when it came to cell type prediction accuracy, and even higher 95% in terms of accurately predicting the spatial Y coordinates of the cells.
Really, better than 9 out of 10 experts.
That's what the data showed for those specific metrics. But perhaps even more compelling was the outcome of a hybrid approach. When they asked SpatialAgent to refine the initial gene panel designs provided by the human experts, it consistently improved the performance of every single human-designed panel across the different metrics. Sometimes dramatically, they mention a maximum improvement of over 900% in predicting Y coordinates for one expert's panel after refinement.
Wow, so the AI wasn't just competing with human experts, it was actually able to enhance their expert knowledge. That really speaks to the potential here. The paper must have delved into how SpatialAgent was able to achieve these results, right?
Absolutely. When they analyzed SpatialAgent's reasoning process for gene selection, they found it assigns important scores to individual genes by intelligently integrating information from a whole variety of sources.
Like what?
Things like leveraging existing reference data sets, consulting external marker gene databases, drawing upon its own internal knowledge from its LLM training, and even considering the information embedded within the human design template panels it had access to.
So it synthesizes multiple lines of evidence.
Exactly. By prioritizing genes that were supported by multiple independent sources, SpatialAgent was able to generate more robust and, well, informative gene panels. And crucially, it also provided clear natural language rationales at the individual gene level, explaining why each particular gene was selected and connecting it to relevant cell types and biological functions.
So it's not just a black box spitting out a list of genes. It provides transparent reasoning behind its choices, which is
Yeah.
so important for scientific interpretability, right?
Hugely important. And the research also looked at how well the design panels preserve the inherent spatial organization of the tissue. Even when using a reduced number of genes, spatial agent selections maintained strong spatial organization and clearly delineated distinct cell clusters that closely mirrored the actual underlying tissue architecture. It outperformed the computational baselines in this regard, too. The top brain genes it identified reflected known functions of that brain region, the dorsolateral prefrontal cortex, and they showed distinct spatial expression patterns within the different cortical layers, which further validates the biological relevance of its selections.
And in terms of efficiency, how did it compare time-wise?
SpatialAgent completed the entire gene panel design analysis in about 30 minutes, which, according to the paper, is significantly faster than the time typically required for manual gene panel design by human experts, and also considerably quicker than advanced computational methods like SPAPROS that they compared it against. So it seems to offer a compelling combination of improved performance and enhanced efficiency.
Okay, that's a strong set of findings for gene panel design. What about the second core use case? Cell type and tissue niche annotation within spatial datasets.
Right, for this, the researchers used a high-resolution molecular and spatial cell atlas of the developing human heart. They compared spatial agents' ability to accurately annotate individual cell types and to delineate distinct tissue niches like anatomical regions against other established computational annotation methods. The annotations provided by the human experts who actually created the atlas, and the author provided ground truth cell type labels.
And what were the outcomes in this annotation task? How did it do?
For cell type annotation, spatial agents' predictions showed a high degree of alignment with the author-provided ground truth. It outperformed other automated tools like GPT Cell Type and Cell Typist, and it performed largely on par with the best human experts in terms of overall accuracy and precision.
Largely on par. Any caveats?
Well, the researchers did note a minor limitation. It struggled a bit to fully recover one specific, less abundant cell type. They speculated this might be due to the LLM's inherent tendency to sometimes default towards more common or well-represented cell annotations in its training data.
Interesting. So even with its vast knowledge base, it can sometimes be biased towards more prevalent information, like a common trap.
That's an important observation, I think. It highlights that while LLMs possess extensive knowledge, effectively incorporating the specific biological context of a given dataset remains crucial. And this is perhaps an area where human experts can sometimes still leverage their nuanced understanding more effectively.
Makes sense.
However, when it came to tissue niche annotation, the task of identifying and mapping different anatomical regions within the heart tissue spatial agent demonstrated really remarkable capabilities. It effectively delineated these regions by integrating multimodal information, including anatomical reference images. And here it performed at a level on par with or even slightly better than human experts in terms of both accuracy and precision. It also showed strong generalization across different regions of the heart tissue.
Okay.
Plus, it offered substantial reductions in the time and, presumably, the cost associated with manual annotation.
So for mapping the physical organization of the tissue, the AI proved particularly adept at leveraging different types of data together.
Yes, especially its ability to integrate spatial information with the molecular data. The researchers also analyzed its analytical behavior and found instances where it made arguably more biologically informed annotation decisions compared to other methods. It did this by considering multiple lines of evidence, like marker gene expression patterns, alongside the overall cellular composition of a region.
Uh-huh.
It also employed what they described as a collective intelligence approach, iteratively refining its annotations across multiple tissue samples to ensure a higher degree of consistency and accuracy.
That sounds like it's learning and improving its annotations as it processes more data. Okay, what about the third key use case? Data pattern mining and the generation of novel biological hypotheses. This seems like where the AI truly steps into the role of a scientific collaborator.
Yeah, I agree. For this, the researchers used a publicly available mouse model of colitis study. And importantly, this study was published after the training data cutoff of the LLM used in SpatialAgent.
To avoid it already knowing the answers.
Exactly, to avoid any potential for knowledge contamination. So, SpatialAgent autonomously executed a comprehensive multi-step analysis of the spatial transcriptomics data. It summarized cellular compositions, identified significant spatiotemporal changes in gene expression, and even calculated cell communication scores using a specific tool called Liana plus Na.
And then,
Following this initial analysis, it generated a detailed scientific report summarizing its findings, and it could refine this report through iterative follow-up questions from the user, as shown in Figure 6a.
and was it able to make meaningful sense of this complex inflammatory disease model? Did it uncover anything new?
The paper reports a broad agreement between the key findings generated by SpatialAgent and the main conclusions of the original published study, particularly concerning the involvement of specific cell types known to be associated with inflammation and colitis, like certain fibroblasts.
Okay, so it confirmed what was known.
It did. But perhaps more significantly, SpatialAgent independently uncovered additional, potentially important biological insights that were not explicitly emphasized in the original publication.
Like what?
These included identifying enhanced TGF-beta signaling pathways, finding evidence of submucosal remodeling in the inflamed tissue, and suggesting critical interactions between fibroblasts and parasites. Based on these findings, SpatialAgent even proposed a detailed mechanistic hypothesis.
A hypothesis generated by the AI.
Yes, regarding the role of fibroblast polarization, potentially driven by TGF-beta and IL-11 signaling, as a key factor in tissue regeneration during colitis.
So it wasn't just confirming existing knowledge, it was actively generating novel research directions based on the data.
Exactly. The researchers highlight the fact that these AI-driven hypotheses, generated autonomously from the data, could have significant implications for maybe identifying new therapeutic targets or even potential diagnostic biomarkers for inflammatory bowel diseases.
That's quite something. Finally, the paper also mentions using SpatialAgent in a more applied setting to customize gene panels for an ongoing lab experiment. How did that go?
Right. This involved a collaborative effort to enhance a standard commercially available gene panel for a study focused on prostate cancer in a mouse model. The researchers tasked SpatialAgent with selecting an additional 100 genes to supplement the existing panel for a specific spatial platform experiment, Xenium 5K.
A hybrid approach again.
Yes, and the resultant hybrid gene panel led to a significantly improved ability to resolve key cellular compartments within the tumor microenvironment, stromal, immune, epithelial. It also enhanced the overall clustering quality of the single-cell data derived from the experiment.
Better resolution, better clustering.
And the selected genes were enriched for biologically relevant processes associated with T-cell infiltration in the tumor, which was a focus of the study. Furthermore, the refined gene panel enabled a more detailed and nuanced understanding of cell interactions within the tumor, particularly highlighting the importance of specific laminin integrin signaling networks that weren't readily detectable with the standard gene panel alone.
So it demonstrated the potential to really fine-tune experimental design to extract even more valuable information from the same experiment.
Precisely. It clearly illustrates the potential for AI to optimize our research tools and experimental approaches for, hopefully, deeper biological discovery.
Okay, so taking all of these findings together, the gene panels, the annotations, the hypothesis generation, the panel customization, what's the overarching significance? What are the potential broader implications of a technology like SpatialAgent for spatial biology, and maybe even beyond?
Well, the paper makes a pretty strong case that SpatialAgent represents a fundamental and potentially transformative shift in how we approach computational analysis for spatial genomics research. It seems to have clear potential to significantly accelerate the pace of discovery in the field.
How so?
And also to empower a much wider range of scientists to engage with and extract meaningful insights from complex spatial data.
How do you see it achieving that broader impact?
By seamlessly integrating these powerful LLM reasoning capabilities, the dynamic execution of specialized tools, and the ability to analyze diverse data types in one framework, SpatialAgent effectively merges the kind of interpretability and adaptability we often associate with expert-driven analysis,
Right.
with the inherent scalability and throughput of automated computational pipelines. This combination facilitates AI-driven hypothesis generation, can provide intelligent guidance for experimental design, and ultimately could lead to much faster, more efficient cycles of biological data acquisition and knowledge discovery.
It sounds like it could really democratize access to sophisticated spatial analysis techniques.
I think that's a crucial point the paper makes. Currently, doing these advanced spatial analyses often requires highly specialized computational skills and, frankly, significant bioinformatics resources.
Yeah, it's a bottleneck for many labs.
Exactly. By automating many of the complex analytical steps and making interpretable decisions along the way, Spatial Agent could enable researchers with perhaps less extensive computational expertise to execute highly sophisticated workflows and still extract meaningful biological insights from their spatial data.
Uh-huh.
It also has the potential maybe to free up computational scientists to focus on developing even more advanced methodologies and tackling deeper, more challenging analytical problems rather than just running standard pipelines.
So it could foster greater collaboration and synergy between different types of scientific expertise, wet lab and computational, for instance.
Absolutely. And the near real-time feedback and iterative capabilities that AI agents like Spatial Agent might provide could also lead to a much tighter, more efficient integration between computational predictions and experimental validation in the lab.
Ah, closing the loop faster.
Right, ultimately streamlining the research process and conserving valuable time and resources. Less guessing, perhaps more guided iteration and experimental design.
It sounds like Spatial Agent is really positioned as more than just another analytical tool. It represents potentially a fundamentally new way of conducting research in this rapidly evolving field.
I think that's really the essence of its potential importance, yes. The paper presents it as a new paradigm for AI-driven scientific discovery, specifically tailored for the complexities of spatial biology.
Well, this has been an incredibly insightful deep dive into Spatial Agent. It's truly remarkable to witness how rapidly AI is advancing to become, well, an active and intelligent collaborator in scientific research.
Indeed. We've seen how Spatial Agent, according to this paper, can autonomously navigate these intricate spatial biology workflows, often achieving performance levels that surpass both traditional computational methods and, in specific tasks, even the expertise of human scientists. And the potential for this technology to deepen our understanding of fundamental biological processes and accelerate the pace of biomedical discovery is truly significant.
So for you listening, I encourage you to consider this. How might autonomous AI agents like Spatial Agent fundamentally reshape the way we approach complex biological questions in the years to come? Could this be a key technology in unlocking the next wave of major breakthroughs in biomedical research? And, what are the potential implications of these kinds of advancements for your own specific field of interest or work? It's certainly a lot to think about. Thanks for joining us for this deep dive.